Today, I'm going to give you some more technical details about codecs. So especially whatever has been used for DNA storage. Okay. In fact, I think today I'm going to focus on whatever we have done actually. I will show you how and what we have done actually in this codec business, because we were anyway earlier contribution. We have done earlier contribution in this. So basically, you have some file, doc file, PDF file, or Excel sheet, or whatever. So now, from computer perspective, you know that you can write programs, so you can find programs at least, which will convert these documents into byte strings. You will have a byte strings document. Now, from that, sometime you want to compress as well. So sometimes, in some of the techniques, you will see that they have used Huffman coding for compression, and otherwise you can use some other compression mechanism as well. So assuming you have done all those things, so once you have the data encoded from, so for example, people have used run length coding here or Huffman coding here, and then they get DNA encoding. So once you have the strings, so now these strings, sometimes people have used binary strings 0, 1. Sometimes they've used ternary strings 0, 1, 2, or sometimes they've used quaternary strings 0, 1, 2, 3. So I'll show you all the three, in fact, today. How, from that string of data, so basically there's a stream of data, and I want to convert into DNA string, right? So what we should do. And once we have the DNA encoding, so we make chunks, and then there is a chunk architecture, which is very, very important. Once we make a chunk of data, so you need to index the chunk. So indexing is very important. Then also, because of some requirements from DNA synthesis point of view, you need to also have some sort of primer sequences at the beginning and at the end of certain size. So there are certain basically requirements from the biology of the DNA, right? So when you want to do things in the DNA writing, so those things are required, right? So then we have this DNA synthesis and sequencing. So synthesis means, you're writing the DNA, and sequencing means you are reading the DNA. So once you do this, and then you store, and then there are errors, right? So errors are of different kind. So I will show you some of the basic things, what kind of errors people have looked at. Initially, people have looked at very simple, so single bit error or 2-bit error, So if there is a string of ACGT and say one letter is substituted. In place of T, you get A. So this is a substitution error. Or it may happen that there is a G which is deleted. Okay, so G is deleted, right? So this is a deletion error of single bit. Or it could be basically, insertion. So there could be, between A and T, a C is inserted. These are the basic kind of operations that are, automatically done in the DNA, right? So they introduce errors. So these are the kind of errors basically. Then uh, most common error is deletion. A chunk of letters are deleted. So basically AAATTC suddenly deleted. Okay, so I will call it as a burst errors in terms of deletion. So bunch of bits are deleted out of DNA. That is another important kind of errors. So how to construct codes which can correct that kind of errors. And by the way, luckily we have coding theory which has developed over the last 60, 70 years, where we know how to correct these errors. And it is because of these techniques and algorithms, we are able to use our mobile phone, computers, and so on, because those techniques are inbuilt, in the system. Okay. And so once you get the data from here, you do the decoding, right? So now decoding, when you do, so once error has happened, you got so and mathematically what we are saying, X DNA is there, which we stored. And when we received Y DNA. So some bits are corrupted with some sort of error. So Y DNA is received, but then we need to convert it back to whatever encoding scheme we have used, whether it is ternary, binary, or quaternary, and then use the error correcting codes inbuilt to correct the errors which was there, right? So I have already told you about the basic coding theory principles, the use of minimum distance. So this is a very, very simple idea that if the encoded data strings have high minimum distance, more or less you will be able to correct more and more errors, using the basic coding theory techniques. But a new kind of coding theory is required. For example, I have shown you Hamming distance, right? Number of places they two strings differ, ACGT strings you can differ, and that can be used. But another very important distance is known as edit distance. What is edit distance? Minimum number of insertion, deletion, or substitution required to change one string X to another string Y. Minimum number of insertion, deletion, or substitution. And this is also precisely a metric. Means it satisfies all the three properties that edit distance between two strings is non-negative and is equal to 0 if and only if X is equal to Y. Number two, they are symmetric. So distance between X comma Y is Y comma X. And third property, the triangle inequality. So all the metric properties are satisfied by the edit distance as well. So it's a very beautiful, and there are in computer science it has been studied a lot. Also in biological applications it has been studied a lot. And there are dynamic programming algorithms to compute the edit distance. Okay. So for example, one can ask you, what is the edit distance between Saturday and Sunday? Two strings. I give you as an example, Saturday and Sunday. What is the minimum edit distance between Saturday and Sunday? Right. So you need to count minimum number of insertion, deletion, or substitution to convert Saturday to Sunday, right? What are those things? That is something. So I was as mentioning substitution, right? So one bit is substituted, deletion, something is deleted or inserted. So for example, something is inserted anywhere. These are the basic kind of errors. So although I would say that since 2012, 2013, how many years? About 9 years maybe, since this field started. And I can see that initially there were only few papers, but now there are papers every week. Okay. So even I find it difficult to read all the papers. There are so many papers are coming, how many you can read. That's why this course I have offered, right? So that now I can also read all of the stuff and then organize the things. So there is a lot of low-hanging fruit actually, you see. Depending upon the kind of error model that you can pick up, you can design an error correcting scheme. And now when you design error correcting scheme, you also know that we need to also keep certain things in mind. The trade-off of the main coding theory problem, you want distance to be as high as possible, right? Or the code rate to be high, right? So if you can construct high code rate, which achieves error correcting capability of certain threshold, of course, in certain error model, that will be a very good and optimal scheme, and then you can actually use it in practice, right? So that is what it is. So I'll show you now initially what we thought the first time when we encountered this problem. As I mentioned, the first paper, there was no error correction. Whatever errors happened, they did it manually, they corrected, because size was less. And later on, there was four-fold redundancy model, which was given by Goldman. And then we used the first error correction technique, which was used Golay support, so I'll talk about that soon. So again, of course, this is the famous paper by George Church from Harvard University. And then this is 2013 paper by Goldman. So, I mean, as I mentioned, these two papers, so there is no error correction here, and there is a four-fold redundancy. So one of the co-author here, Emil, she founded the Twist Biosciences company, it's doing very, very good actually. So there's a whole DNA storage division, so this paper is very interesting. These are two initial work. Okay. After that, Robert Gross is also doing a lot of work in, he is expert in data storage mechanisms, especially magnetic beads and so on. And, in Europe actually. So he has done a lot of work. In fact, glass-based data storage techniques they have come up with, and he has used a code which is known as Reed-Solomon code. I'll tell you briefly what is Reed-Solomon code. For massive data storage applications, Reed-Solomon code has been applied to all kind of, your QR code is based on Reed-Solomon code that you often scan, your CD, DVD, high definition TV, your telecommunications, deep space communication, communication with the Mars or Jupiter or whatever. It's very powerful, beautiful, uh, you know, mm, error correcting code, which is known as Reed-Solomon code. In fact, there is a whole book, Reed-Solomon code by Stephen Vicker, and there is another person, I forgot the name. He's an Indian person actually. Those two people wrote that book, Reed-Solomon code and its applications. You can really want to see. And actually, the Reed-Solomon codes uh, are designed in such a way, you take the message bits, okay, and you work on some field. Field will say 256 number of elements. Say Galois field with 256 number of elements. So G of 2 raised to power 8, right? So of course there will be some working polynomial to construct the field and so on. Now what you do, you take the message bits. From message bits, k message bits, you form a polynomial. First message bit M0 plus M1x plus M2x plus dot 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 M k minus 1 x raised to k minus 1. So there is a degree k minus 1 polynomial from basically message bits. Once you have this polynomial, then you take certain points on the field and evaluate this polynomial at those points. Okay, so the code word will come at these points. So C0 will be M of evolution of this polynomial at that particular point, say alpha naught, and other one will be at alpha 1, and so on. Like that, the field elements you take and evaluate the polynomials, and that's how you get the code words. It's a really beautiful construction. Nowadays, people don't talk about this much. This is the original way how these things were constructed. And in fact, I don't know whether you heard about RAID. Have you heard about RAID 1, RAID 2, RAID 3, RAID 4? You have heard. Where? Operating system. So all these are based on error correcting codes, if you see closely, right? Patterson gave this idea quite old, I think around 70s or something, So it's a really nice RAID 1, RAID 2, how it is based on Hamming codes and so on. In fact, Reed-Solomon code based approaches are also used in certain RAIDs, in the recent ones actually. It's really beautiful. But now actually, as I mentioned, there is a polynomial based description which can be, which can be used to give you the idea of Reed-Solomon code. So I'll talk more about when we'll talk about Reed-Solomon codes, but I just wanted to mention that this is very, very important. So Reed-Solomon code is very, very important. So you should know, how to construct Reed-Solomon code, okay? Anyway, so they have used this Reed-Solomon code initially. Then I think a lot of work was done by Olgika at UIUC. So she has given some initial error models, and you use different kind of distances defined on the DNA strings for correcting the errors, by looking at different error models. So I'm going to talk about those things later on. And then of course there are many. So you see, you pick up any classical coding theory idea and see whether you can construct DNA, storage model, based on the errors and how much you can take further. For example, low density petri check codes, LDPC codes, all different kind of coding techniques people have used. Okay. So that's all. So basically, before that, this is a really elementary work, which is okay. I think even when we started doing these things in 2013-14, there were only about 7 or 8 patents in this area. Now there are many, by the way. Very often people are filing patents, especially Microsoft and all these companies are filing a lot of patents. But still, I would say that there is a lot of scope in doing some things and filing patents. So if you are really interested and you can do something meaningful, that can be done, I think, out of this course. So I'm going to today talk about, so of course, this paper is just merely implementation with the, little bit of modification with the Goldman paper, which was implemented by Shalin Shah, the DNA cloud. Okay. But I will talking about another paper, optimal family of codes for archival DNA storage. So here I will show you how we have used, because initially when this problem came to us, so we were not sure which error correcting code we should use and how, right? So I will show you how we have thought about it and how to, I'll show you that correction, and I'll also briefly talk about another work that we have done with Professor Vaneet Agarwal at Purdue, And then I'll talk about briefly from some papers from here. This is the initial set of papers. Now I cannot list, the list is very large, but anyway. So, okay, so there are two kind of models for DNA storage. One is archival DNA storage. Archival DNA storage means you store data into the DNA and you forget about it, right? It's in the test tube or maybe dry DNA, whatever you can make a powder that is enough to store data, right? So archival data storage. So techniques for error correction in archival data storage and techniques for error correction in rewritable DNA storage is pretty new, and some progress has been made, but still a lot of challenges are there. People are working on it. So you know that of course we will be able to use DNA storage just like a pen drive. You insert and data is written, and then you read and write as many times as possible. That technology is not yet available, but it will be available soon. People are working on it in different ways because it requires hardware. It requires understanding of the chemistry, how fast you can do the synthesis, means DNA writing, and how fast you can read the DNA. Reading DNA has been designed a machine like pen drive by Oxford Nanopore sequencing technology. I'll talk about those things in details, how that sequencing works. So basically, that has come up to, because sequencing is an old problem. People are doing sequencing since last 20 years or so. I think started in 1989 or 1990s and so on. And now we have, as you know, that Eric Lander did a lot of work. Now we have robotics, so robots can actually do faster sequencing. There are machines, different sequencing machines are there for reading the DNA, because we want to understand what is the DNA of a tree, or maybe, a flower, or maybe any fruit. You want to understand the DNA, or any species, right? So you want to read the DNA first. So that's why reading technology has been developed a lot, and it has come up to the pen drive kind of thing. But for the synthesizer, is still a challenge. So people are working, as I mentioned, Twist Biosciences and other companies are working towards, DNA printer, so you can have fast writing as well. So once you have fast reading and writing, then only you can actually have a technology which you can insert into your laptop and do the things. Anyway, so now for this archival DNA storage, we have data source, then we have encoding, DNA coding, and of course all the basic things. So now I'll just first of all tell you what is the DNA code. So basically, when we are saying that we are going to have data stored in DNA strings, right? So these strings of ACGT will come from some space, right? So this space is sigma DNA. So sigma DNA is, suppose, ACGT, adenine, cytosine, guanine, thymine. These four things, they work as a alphabet for DNA. So sigma DNA is having these four elements, ACGT, and our DNA code is simply a subset of sigma DNA raised to power n of length n. So length is n, total number of code words capital N, and minimum Hamming distance D. Exactly, quaternary set is whatever we have done in the quaternary coding theory can be copied here directly. Just a simply subset. That is nothing but a DNA code. So what is our main objective here? Minimum Hamming distance among the DNA strings, that where we are storing the data. So actually it's all rough. I would say that people assume that if the Hamming distance is at least this threshold, we know that we can correct these many errors, while they're doing. I'll show you in our work how it works actually. But people go more about edit distance, because in practice, when you do the experiments, error will not follow one specific model. Okay, I will follow this model. Error will, this time I will follow that model, right? It is something that happened, right? And you want to control the errors, right? So with sufficient reasonable way of, if you can error model can capture what are the possible errors. And then as an engineer, what people do, they simulate millions of data set and just make sure that it's working. Maybe, in millions, only one bit is corrupted. Okay, that chance is, you can take it further, right? I mean, you can design the system as an engineer, right? Mathematician will say that, okay, no, I want a bound, right? That's something different. But engineers will go away with that, okay, so that is the thing. So let me show you the main objective. So roughly, so we say that we want to design optimal DNA code with maximum minimum distance to correct more errors, right? This is we want to design a collection of DNA strings of ACGT of say length n with maximum distance possible, and capital M is as large as possible. Similar coding theory constraints we have put it here, right? Also, we want maximum storage capacity achieving code. So there are two kind of information density we can talk about. One is physical density. So we know that 1 gram of DNA can store 455 exabytes of data. Okay. So can we really reach in that order? Can we really go and build that system? Recently, George Church, this year only, in fact, was one of the reviewer of that paper that appeared in Nature Computational Science, where they have actually designed a codec called Yin Yang codec, right, based on Buddhist philosophy, which achieves something around 432 exabytes per gram of DNA with the experiment they have done. This is a great achievement, right? You see, so now they have done, but it costs a lot of money, right? But the thing is that now people will further come up with more improvements and so on, right? George Church, same person who started this area in 2012, now has achieved that kind of capacity in reality. That is the physical density. Then there's the information density. How much bits you can pack in one DNA bit, right? So how many information bits you can pack? That's another density, right? So the ideas of Shannon capacity will come into picture. So I'll talk about those things more in details. But so basically, we want maximum storage capacity achieving codes, right? And which can correct as many errors as possible, right? Then it will be good code, right? As an engineer. As an engineer, it is also important to design the chunk architecture of the DNA blocks. So normally, when we design DNA codes, I mean DNA strings for storage, our current machines, it's the DNA molecule is more stable if you go up to 100, the size of 100 nucleotides, so ACGT of size 100. If you go longer, chances of error will increase, right? So normally people will take 100, 200, sometimes even 300, but it depends on which machine you are using for writing the DNA or reading the DNA. So 100 is a very good size. So actually, in that case, that means people want to design the architecture of the DNA in such a way that there is information, there is a chunk architecture. In the chunk architecture, there is an indexing of the chunk and some other requirements which are coming from the biological constraints. For example, primers, which will start the reaction and so on and so forth. All kind of things will come into picture. So you need to design the chunk in a really nice manner. So that is also one problem for engineers, how to design efficient chunk architecture. Okay, depending upon the current size of constraints given by the machines, like, okay, 100 nucleotides are good enough, or 120 or whatever, some small number. Okay, so this is also another important thing. So if you look at the first model by George Church, Gao and Kasuri model, we have data source. Here they've used run length encoding. So here I would like to give you your first T problem for today. What is run length coding? Okay, what is run length coding? Which I have used here. What is run length coding? At least you should understand a little bit about it. What is run length coding? Okay, it's not very difficult. Since you know the coding theory, now if you look at what is run length coding, you will understand. Okay, what is run length coding? Of course, now so once I have given, I may ask in the exam also. So you should know what is run length coding, right? Okay. So then we have DNA coding. This is the standard synthesis sequencing and decoding. The chunk architecture was information bits and index bits initially, and length of the chunk was 115 nucleotides. Number of chunks was this when they stored a book of about 2.4 MB or something like that. When they did the first experiment, and there was no error correction. So this is but you see, this paper came into science because the first time somebody is doing it at a large scale data storage into DNA and retrieving it back, right? So this is something interesting. So in the church example, this is just a symbolic representation. I mean, it's not an actual example, but I'm just showing you. So say DIACT, ST values are these, binary values you can find out accordingly, right? The 19 bits barcode appended for DNA block identification. So some random block barcode I have assigned here. And then this kind of encoding is used, 1 for T and G, 0 for A and C, kind of thing, okay? And so once you have that, 22 bits primer sequences are added. These green ones, the primer sequences were added for DNA synthesis, reading the DNA, writing the DNA, okay? In fact, I will talk about it in the course more about primers is a very important. How do you design efficiently primers? That's also interesting mathematical problem as well as engineering problem. Because if error occurred on a primer, it's much more problem. So your whole architecture will go away, right? So you don't want primers to be, they should be error free. So how to design error free primers? That's another interesting problem. Anyway, so in the Goldman model, so Huffman coding. So you know from discrete math course, I don't know whether you remember, we talked about binary Huffman coding, right? So this is ternary Huffman coding, some generalization of that. And then we have information bits, index bits, and a parity bit. Okay. The length of chunk was 117. Number of chunks was these many. Fourfold redundancy added for error corrections. So this is the paper that appeared in Nature, and this was the paper which I gave it to second year BTech student Salin Shah in our class. And in one of the summer he came to me and I said, this is the paper, just implement. So he implemented the algorithm and then realized that there is a problem. This algorithm, though appeared in Nature, but it can only encode up to 500 MB file. After that algorithm there was a problem because the chunk architecture was fixed. This was a drawback of this approach. So we did variable architecture in the chunk, and that has given us much more flexibility. So then we were able to code larger files, even 1 GB, 2 GB. Okay, so that was the thing. But anyway, so this was the improvement on the algorithm. So that's why when you want to start something, take this paper, implement it, then you realize what are the things, and then you can do the improvements. Now, again, I think in this one Goldman example, DICT is ST values, ternary Huffman codes are written. Couple of other things I am showing you may not be accurately, may not be very accurate, but anyway, I'll show you how this is done. I think length is here written it is 30, but you can see that S1 size is 36, right? Some errors are there, but anyway, some padding of zeros and so on. So we have this. So this is a beautiful thing actually, and this map I think I mentioned to you earlier also. What it does, it takes, so here in this example it is taking ternary bits, right, 0, 1, 2, and giving us a string of ACGT such that no two ACGT are consecutively occurring there, So AA, CC, GG, TT will not occur anywhere when you do this mapping. So this is the property of this mapping. Okay. So we focused on this map initially, and then we have, there are many interesting properties. After that, we worked on this map further. So this is the mapping which is making sure that things are not repeated. Okay. So if you are interested in working more, we have some other interesting problems related to this map. I will talk about it in the course. What are these interesting maps? So this is we call it as non-homopolymer map, right? So homopolymer means a string where ACGT repeats, AACC repeats. Non-homopolymer map means this homopolymerity is not there. So basically, AACC, GGTT will not occur. Okay, so that is a property. So how it works? So you can see that you have 1 there. Assume that previous nucleotide is A, 1. So we'll get G here. You can assume anything out of here. You can assume A, C, G, T, the previous nucleotide, and then you start. So if it is G here, you see that one is G. Next one is V. Next one is 1. So you again see 1 and G is A. So you get A there, right? Like that, you keep on moving, right? So this is a very interesting map. In fact, I will show you how we exploit one property of this map to correct 2-bit errors in the DNA. So then chunks are made of length 100 and so on, Identifiers are, indexing is done for the chunks and so on. Pairs are added. So this is all pretty engineering stuff, So let me show you. Yeah, so Goldman encoding model. It uses 4-fold. So we implemented this Goldman one, this software. So I'm going to now talk about this work that was published in IWSDA conference. By the way, both of these papers, see that the DNA cloud paper and this paper, is the very early work, So Madhav, as I mentioned, Madhav was in LinkedIn. I think I don't know where now he is. Vijay is in ISKCON temple, right? From Sadhu, right? So he's there. ISKCON. Or Dikshita is in London actually, working in a company. So I'm going to discuss these two things: DNA Golay subcode model and constraint code model. Okay. So first, let me describe DNA cloud model. So both are. So this was done by by all by each of us, and then I think this is So the constraint code model was done by in collaboration with Professor Vanit Agrawal at Purdue University. So when we published that work, then Professor Vanit Agrawal contacted us, and then we started collaboration on the second one. So let me show you. So I gave you till now one problem to work, right? I will give you a few more. So remember, these are important problems. So this is your, Goldman model. So I'm talking about this paper actually. So this is the paper. The technique that we are going to use is the distance. We will use distance for correcting more errors. You will see normally if distance is d, you can correct d minus 1 by 2 errors, right? And somehow, because of the property of the map, we are able to correct more errors than required. So let me show you. So what we did, you remember the byte strings. So we want something, the total number of code words should be 256 because the ASCII values are 256, right? So we looked at 256 number of code words of different length and what is the maximum distance possible. I looked at, these are all ternary codes. Remember I showed you once in a class the tables, right? So I looked at the tables, which one are the best possible codes for the ternary. So we listed these 1, 2, 3, 4, 5, 6, 7. 7 of them. Which one to use? It's a question, which one to use? Which one is the best one? So I will show you why we use this second one. The reason will be clear to you. Sure. Because you see that the first one will correct single error, second will correct 2 errors, distance is 5, 7 will correct 3 errors, and so on, right? As you move along, you can correct more and more errors. Why not use that one or this one, right? So you also want to minimize the length also, right? If this is too big, then there is a problem, right? So this is, the best thing is, this code is the best rate actually, So rate is the good one. So we used a subcode of, so this is 11.256.5 is a ternary Golay code. So basically it's a subcode of the ternary Golay code. I'll show you what is ternary, what is Golay. Golay code is a beautiful code, very beautiful code. And I will show you the paper that he wrote is just 1-page paper. Okay. And it became a very famous paper. Mathematically, it is very beautiful. From application point of view, it is very beautiful. And also, it is also beautiful in terms of, lot of other, it is also corrected. It's a perfect code actually, first of all. it meets a sphere packing board with equality. So that is another very interesting property. So there are only three perfect codes: repetition code, Hamming code, and Golay code. And Golay code is something, how people found about it. They looked at a sphere packing bound, and they look at the parameters of NMD. They try to enter NMD parameters and see when equality will happen. So one case was repetition code, another case was Hamming code, the parameters, and third parameter was this. Then they said whether there is a code, how to construct. That was a challenge. Nobody knows how to construct a code with certain parameters. It was Golay who just wrote one-page article, and he constructed both ternary and binary Golay code nicely. So which was okay. So important thing about this, our construction, this Golay code, with the help of this mapping, non-homopolymer map, we are able to correct 2-bit flip error in the DNA, at the DNA level. So I'll show you how that works actually. It's really interesting. And the capacity, of course, this is all theoretical. So we arrive a capacity of 115 exabyte. So it was a good thing. At that point of time, Goldman was 2.2 petabytes. And this was in exabyte already. So this was the first exabyte scheme, that we presented actually, in that order with correcting 2-bit flip errors, per gram of DNA. Of course, we have not done any experiment, so that is there. So we also saw, I will show you the comparison with so Goldman has taken 5 files, they have encoded from their scheme. So we have taken the same 5 files, image and PDF, whatever, and we have shown our comparison that how good our scheme is actually in terms of error correction, in terms of even amount of DNA that is required, Key idea is that our architecture is variable length architecture. This is very, very important. We picked up this 11.56. So now this is a paper by Golay. One page, this much, write up. See? So he finished one interesting open problem just by writing this. Okay. So I hope you can see their thought process. Where is mathematics? It's all writing in poetry, right? And then there is a matrix given, right? So analysis, this is how people think about it. Anyway, this is the binary Golay code. By the way, both the Golay codes can be constructed. So they are beautiful things. So they can be constructed from beautiful things. So as you know that mathematics is queen of all sciences. So number theory is what? Crown. So the queen. And quadratic residues are gem in the crown. I mentioned this earlier. These quadratic residues can be used to construct this Golay code. And both. See how? Let me show you. So this is, 24. I should mention this is 24, 12, 8. Length is, this is binary actually. Length is 24. Dimension is 12. Distance is 8. If you look at literature on Golay code, you will find different applications in mathematics, lattice construction, all kind of things, beautiful things. Anyway, so you can see that there is an identity matrix. This is I11, identity matrix I11. And then, one more here. So overall I12 is there, right? So all these rows are linearly independent, basically, because of the identity matrix. So you will get 2 raised to about 12 code words, right? 2 raised to 12 code words. Now, look at this part, how this is constructed. Here there is all one vector. Now, if you look at this 0, 1, 2, 3, 4, I am just numbering them. So there are 11 of them, 11 columns at the top, 0, 1, 2, 3, right? So apart from 0, so at the place of 0, I am putting 1. This is a notion you can think of. Now 1, 3, 4, 5, and 9 are the places which are quadratic residues modulo 11. P is 11 is a prime number. Modulo 11 So basically 1 square is 1 modulo 11. So this is a quadratic residue. 3 square is 9 quadratic residue. 4 square is 16. Modulo 11 is 5. Right. So all these are quadratic residues. So basically 1, 3, 4, 5 and 9 are quadratic residues. All other places are quadratic non-residues. So all other places it is 0. In place of 2 we have 0. 6, 7, 8 and 10 we have 0. All other places it is 1. And then what you do, the next row is a shift of it. Cyclic shift. All rows are cyclic shift. And you arrange here. Beautiful. There's a lot of symmetries. Now you can argue that the minimum distance of this code is 8. How? See that this portion will contribute 1 from here and 1 from any of the diagonal entry. So 2 bits will will contribute, right? In the distance, if you want to compute the distance between any two vectors, right? And if you look at here, so if you see that, take first 2 rows for example. So they will share how many? 1, 2, 3. 3 ones, right? So basically, if you take any 2, so basically how many of them are different? 1, 2, 3, 4, 5, 6. Any 2 rows will differ in 6 places. Beautiful thing. So 6 here and 2 there, 8. Now you have to just show that, you cannot go below 8. The 8 is the minimum distance, It can go above than 8, but not below than 8. So it's beautiful. So just by looking at the matrix, just observe, you can find out different properties of this Golay code, right? So, of course, you can give a couple of lectures on Golay code. There are different decoding methods, efficient decoding methods, connections to geometry, connection to lattices, connection in applications also. People have used Golay code at many places. Okay, so this code 24.12.8 is known as extended binary Golay code. Extended binary Golay code, and the distance is 8, so you can correct how many errors? 8 minus 1 by 2, right? Those many errors it can correct, right? Distance is 8. If you actually puncture it at any column, maybe say last column, so you will get a code of length 23, dimension 12, and minimum distance 7. That is the actual Golay code which is fitting the sphere packing bound parameters. Okay, this is the extended version of it. Okay, both are beautiful anyway. So this is all about binary Golay code. So first I looked at this. I thought maybe should we use this for the DNA storage? Maybe you can still use it. I don't know. I have not tried after that. Maybe you can think of, can we use this? Something is beautiful, should have some beautiful applications, right? So you can always try doing something here. But we looked at the ternary one. So ternary Golay code over the field Z3, integer modulo 3, entries are 0, 1, 2, right? So this is the identity matrix and some matrix B here. This can also be constructed from quadratic residues. Okay. So I leave it as an exercise for you how the ternary Golay code can be constructed from quadratic residues. So you can figure it out, Okay. Ternary Golay code can be constructed through quadratic residues. Okay. This is something which you can figure it out. Okay, so now we have 12.6.6 ternary Golay code. You puncture one column, you get 11.6.5 ternary Golay code. Total 729 code words. We looked at all these code words carefully, and we selected certain code words out of it for our purpose. Okay, so for error correction. So we are using basically a non-linear code. So this is a linear code, right? You take 2 code words, the sum will be in the set. So we actually looked at non-linear code. You see, this is the list. This is the subcode, and we mapped it to all 256. So basically, we took 256 out of 729. We took 256 vectors, and we mapped it to the ASCII values, and we make a table. We made a table, and this table we use for encoding. Okay, this is what we did actually. Now, clearly you can see the distance is 5. So we'll be able to correct 2 errors without any problem, right? Even if it is non-linear, no problem. We can correct the errors. But actually using map, we are able to do more. So that's what I'm going to show. Okay. So chunk architecture, we designed, certain info bits, chunk header, base pair for index and so on. This is Goldman encoding, modified. I think this we already talked about it earlier, so I'm just skipping these slides. We have this ternary code words, S1. We gave you this lecture in the first lecture, right? So if you remember from there, we have done this calculations, right? File extension and so on and so forth. Everything should be a multiple of 99. This is, and again, there is a map that we used to construct this ACGT. Final strings, chunks of length 99. Indexing the chunk, we have these red, the pink things are added towards the end, right? Now, just observe this. I think I'm about to finish in 2 minutes. So this map, has this property. So what is, first of all, this is not a unique map. There are many other possibilities. One map is this, another map is this. You can either use this or that, or there are any other possibilities. So you can use any of the map. It's not unique. That is this lemma. Okay. Next. If X DNA from cDNA is sent through a noisy channel, and suppose Y DNA is received. So we stored X DNA and Y DNA is received, right? If the distance between these two is T, its T errors has happened, suppose, T bit flips, I'm assuming, okay? Then the corresponding, suppose, the corresponding ternary code obtained using these are, suppose, X comma Y. So you go back, X DNA was stored, Y DNA is received. You map both the vectors back to the ternary space, right? Because ternary is a Golay subcode, right? And in the ternary subspace, the distance cannot exceed 2T. If that distance is T, this is the property of this map. Okay. If the distance between two vectors or two DNA vectors is T, the corresponding ternary vectors have distance at most 2T. Okay. So this is the key property. You can see that if X DNA is this, Y DNA is this, the corresponding, vectors are like this, and the distance is 4 here. If this distance is 2. Okay. Should we do it today, or because otherwise you will see? Maybe, okay, we'll repeat it again, okay, next time, right? Because then I probably will take more time anyway. We will talk about it in the next class then. Fine. So that's all for today. So just remember whatever I have told you, that we had run length coding. I gave you something about Golay code also to verify, right? Those things will give you more idea. We cannot cover a whole coding theory, but at least few things which are needed, you can always go back and look at it in details, right? Fine. So we will do more things in the next class then. Okay. Thank you.